I end well. But I don't know because they went up to here. So I'm ending where here. You're ending this slide. You know, it doesn't show the slide number. Let me just say I'm ending at the types of microbial growth media. Yeah, where you come on top of the examples of different. Yeah. So the moment you reach here, you also see this. So you see you're stopping. You'll see this card in this box. Yeah. You just bring it back. So I can begin. Yeah, a big bag. The other small thing, yeah, the my how much do they cost? Yesterday I went to Konyo. I thought I would get one, but I failed. This one that's from Tanzania. Yes. Good morning, everyone. How was your weekend? This morning, we are going to talk about microbial growth or how microorganisms can grow. When we talk of microbial growth, we are going to look at microbial cultures. These are media where microorganisms can be grown so that they can be investigated upon, find out the specific microorganism. Okay. Microbial cultures. These mostly are made of microorganisms. They are made of microorganisms. And the microcultures, these are methods of multiplying microbes by letting them to reproduce in certain culture medias under controlled laboratory conditions, so that the individual or the laboratory technologist or assistant is able to determine the type of microorganisms. For example, is it a bacteria? Is it yeast? Is it mold? Or are they pathogens? Okay. So microbial cultures, they also help to determine the balance. They also help to determine the abundance or counts of total microbes or specific microbes in a sample, maybe a sample of urine or a sample of blood. Then microbial cultures also help in isolating various or specific microbes. Let's look at the growth of microorganisms and what they require for their growth. Because for microorganisms to grow, they will require certain other items or medias. So microbial growth refers to an increase in cell numbers, for example, an increase maybe in bacteria, an increase in yeast, and an increase in complexity of structures, for example, in the molds, So microorganisms and the growth requirements, these are commonly represented as cells per unit mass or the volume, or how many cells have grown per unit mass, or how many cells have grown in volume. So cell numbers are often represented as log 10 since they are larger. Bacterial cells, remember we said the other time that they reproduce by the process of binary fusion. So the time taken for cells to double is called generation time, or the time taken for cells to divide from cell formation to division. At times, this is also referred to as generation time. So the time taken for cells to double, it varies from species to species. When we talk of species, it refers to the species of microorganisms and also the conditions and also the growth condition microorganisms have been subjected into. So microbial growth, we have a growth curve that will show us the phases of microbial growth from inoculation into a growth media. When we talk of what we mean by inoculation, it means the introduction of the infective microorganisms into a particular media. So we have the following phases. We have the first phase, which is known as the lag phase. Second one is known as the log phase. Third is known as the stationary phase. Then the last one is known as the death phase. So when you look at that curve represents the growth and the growth media of microorganisms. You can see A is representing the lag phase. Then you can see in B represents the log phase. Then you can see in C represents the stationary phase. Then D represents the death phase. So let's look at what happens in the lag phase. That is the first stage. In the lag phase, this refers to the initial delay. It refers to an initial delay in growth following inoculation into a new growth media. Remember, I told you that inoculation means the introduction of the infective microorganism into a certain growth media. So when this microorganism is introduced into the growth media, it means that there will be a delay in its growth. Then also microorganisms or microbes will have to adapt to the medium and synthesize enzymes needed to break down the substances in the growth media. So this tells us why indeed there is a delay in the this tells us why indeed there is a delay in growth, because the microbes have to adapt themselves into the new media they have been put into. The second phase is known as the log phase. In the log phase, this is the stage of rapid growth or rapid reproduction or logarithmic growth. What happens in this log phase is when we plot, this is a plot of the log numbers of the cells versus time gives a linear relationship. What does it mean? It means that the cells are at their greatest activity in this phase. That is the stage of what? Activation. In other words, in the log phase, the cells are growing at a faster rate. So this stage is preferred when transferring culture into fresh media, since active cultures rapidly dominate new environments. For example, if you want to transfer a culture of maybe yeast into another media, you are going to do it during the time of the log phase, because this is the time when the culture is growing very fast. Stationary phase. In the stationary phase, this is when the available nutrients are depleted or toxic wastes accumulate. Remember, as the microorganisms continue to grow, they also utilize some of the nutrients. So when they utilize the nutrients can get finished. Or at times they also release what? Toxic products, which will also accumulate. So an equilibrium phase, this is where the growth rate is slowed down and the cells are also dying off. No net change in cell numbers. Okay? Remember, when the microorganisms grow, then time comes when the growth rate will slow down. Then when it slows down, then the cells eventually start dying off, which means that when the cells die off, it means that there is no net change in cell numbers. The death phase. In the death phase, the formation of new cells ceases as existing cells gradually die off. As the word say, as being referred in the sentence death phase, it means that the cells have died. And once they have died, then it means that these cells do not exist anymore. Remember, the log phase can be made longer. The log phase can be made longer by removing some of the toxic wastes and adding more nutrients, or both. Because when you add more nutrients to the microorganisms, then it means that these microorganisms are going to grow because they have been given what? Food that requires them to survive. So in the log phase, you can make it longer by either removing the toxic wastes or adding more nutrients, or both. That is, if you want the log phase to be a little bit longer. Preservation mechanisms prolong the lag phase, for example, cold temperatures and some chemicals.
You use this.
Okay.
Yeah.
Let me try to go back where I was. When you press like this, it doesn't go. Maybe it needs me to use a lot of force. because I did like this, it was not moving. So maybe it requires a lot of force. Think. Okay. We'll use a little bit. When I click this tab, if I use this tab to go,
What is it doing?
if I use the tab to click there,
To click what?
to click so that I go to the next slide,
Mhm.
it goes very, very, very fast. Then if I'm using this, at times it doesn't move, then at times it will move.
When you use it.
Hmm?
Use it.
I think when this data is here,
Which one?
Pues se puede ir.
No, maybe I used that.
on storage system.
And you end?
Yeah.
The thing is, what actually your challenge is here.
Where?
When you this thing of yours reach here,
Mm-hmm.
so when you press next, it's going like that.
Mm-hmm.
It doesn't mean that you're gone. It's just coming back here. It's the same slide. But this
Never itambula. Esigala ide.
Esigala yemu.
Inagenda maaso.
I didn't know. For me, I thought it goes so fast.
Kamera kuri bulungi.
The camera is okay.
Okay.
Mm.
Yes, like that.
Okay.
Hey, no way kaduka with that.
when you come here, you press next, it runs like
Let me try to press again.
yeah, you just press. Yeah. That's running. Make sure it's still on the same. The pointer.
Then I can be using the pointer.
Just press here next. If you want to come back, you press this arrow. If you want to go from here, press this.
I think the pointer is better. Let's continue with some of the factors that affect microbial growth. Oxygen supply is also another factor. When we talk of oxygen supply, this oxygen supply can be in form of aerobic or anaerobic conditions. Then we have acidity or pH. Then we have the redox potential. Then we have the presence of natural antimicrobial substances. The first factor affecting microbial growth, which is temperature. Microbes can grow very well within a temperature range, for example, between freezing point to when protein or protoplasm coagulates. Also, microorganisms will grow between the maximum and minimum points. Between the maximum and minimum points, this is the optimum temperature at which organisms will grow best. Below the minimum point, it means that the growth of the cells stops, but the cell is not killed. Okay, the growth of cell stops, but the cell is not killed. Then above the maximum, cell death occurs automatically above the maximum. It's so hot. So if it's really, the temperatures are very high, it means that the cell will automatically die off. Most cells die on exposure to heat. For example, temperatures of 60 and above or 70 degrees for 15 seconds. When you expose the cells to high temperatures, for example, 60 to 70 degrees centigrade for 15 minutes, for 15 seconds, sorry, most of the cells of the microorganisms are going to die. However, bacterial spores require more higher temperatures, or they require more severe heat, for example, 120 degrees for 30 minutes. Usually the temperatures that are found in the autoclave, they are very high. That's why an autoclave is the best equipment for sterilizing most of the medical items, for example, the theater clothes. So bacteria can be classified also according to the temperature preferences into 1, psychrophilic. The psychrophilic bacteria will be able to grow at temperatures below 16 degrees centigrade. Then we have the second category, the mesophilic bacteria. This will be able to grow best at temperatures between 16 and 40 degrees centigrade. Then we have the thermophilic. Thermophilic, this grow best at temperatures above 40 degrees centigrade. If you can have a look at that diagram, you can see the psychrophiles and the temperature range. Then you can see the mesophils and their temperature range. Then you can see the thermophils, their temperature range. Then you can see the thermophils with their temperature ranges. So the graph has been plotted. We have the growth rate against temperature in degrees. Let's look at the other component which affects the growth of microorganisms. We have nutrients. Microbes require nutrients for growth, and the nutrients requirements, they differ. Remember, microorganisms now are also like what? Maybe if we compare them also to human beings, because human beings need what? Nutrients to grow. So the same. Microorganisms also need what? Nutrients for their growth. However, the nutrient requirements also differ. For example, when the microorganisms need a source of energy, they need carbohydrates, nitrogen sources, minerals, and also vitamins. So lack of any of the essential nutrients inhibits or slows the growth of microorganisms. Depletion of the nutrients also slows down the growth of what? Microorganisms. Depletion, it means that you have removed some of the nutrients that are required for the growth of microorganisms. Water or water activity, microorganisms also need water as a medium for them to grow. Many organisms quickly die off under dry conditions, while others tolerate for long. For example, bacterial spores can survive for so many years. For example, the bacterial spores that cause tuberculosis, they can survive for so many years. Water activity is used as an indicator of availability of water for microbial growth. So generally, cell growth ceases when the water activity is less than 0.8. Oxygen. Oxygen is also another component needed for microbial growth. So the oxygen requirements for microbial growth and survival, they differ. You'll find that some microorganisms require oxygen to grow, and these are known as the anaerobes, the aerobes, sorry. Then some do not require oxygen because oxygen is toxic to them, and these are known as the anaerobes. Some can survive in the presence of oxygen or absence of oxygen, and these ones are known as the facultative anaerobes. An example of this is the E. coli. We have the group A streptococcus. Acidity. Sensitivity to pH varies from one species of microorganism to another. Some microorganisms have a minimum, optimum, and maximum pH at which they can grow. But most microbes have optimum neutral, optimum near to neutral pH of around 7. Other organisms can tolerate pH which is very low, and these ones that tolerate pH which is low, they are known as acidic. They are known as acidic. An example is the lactic acid bacteria, which can grow in fermented products. For example, the yogurt at a pH of 4.6 below. Let's continue with the microbial growth media or culture media. Culture media. When we talk of culture media means a nutrient material that has been prepared for growing microorganisms in the laboratory. These special media are designed, or they have been prepared to support the growth of different kinds of microorganisms. So these culture media are composed of different nutrients that are vital for microbial growth, imitation of natural substances. In other words, these cultures, they are an imitation of some of the natural substances that will facilitate the growth of microorganisms. So the culture media come in different types. Why are they coming in different types? This is because there are various microorganisms, and the various microorganisms each have a unique property, and each has a specific nutrient that will allow it to grow. So the differences in media also depend on what nutrients they contain and what function they play in the growth of the microorganisms. Examples of culture media. Culture media can be in a liquid form, or you can call it broth, or they can be in solid form. You can call it agar, or addition of water to agar forms. When water is added to agar forms, a gel will form a solid media. So microbes will generally grow on practically any source of organic food which provides carbon compounds. Remember, carbon compounds, these are the ones that are responsible for giving glucose. Okay, so microorganisms will grow in any compound which has what? Carbon, and these carbon compounds will be able to give glucose. Nitrogen compounds are also found in some of the proteins for the microorganisms to grow. And the main sources of nitrogen compounds are, we have the peptones. Peptones are got from partially digested proteins, vitamins, minerals, and also inorganic ions, water. Microbiological media are also formulated to supply the above. In other words, the medias that are used for culturing microorganisms, they have been created in such a way that they supply the microbe with what they need to grow. So in the laboratory, we have two categories of media that are being used for the growth of these microorganisms or culturing microorganisms. We looked at the broth. Broth is a soup-like, and it's in liquid form, and quite often it is prepared in tubes. Then we have the agar media. This is a set of jelly-like substance. It is formed by addition of agar, and the agar is a seaweed extract at about 15 grams per liter. It is melted and poured into glass. It can be recycled or non-recycled. So it's put in either, it's poured in a glass containers. So if it is poured into glass containers, it means it is recyclable. It can be recycled easily. If it's poured in plastic containers, it's not recycled. Or containers like petri dishes or plates can be used. Agar. Agar is a complex carbohydrates, and it's derived from the red algae, for example, the chondrus or Geridium. So this agar is supplied in a powder form, and it must be melted to mix it with the water. Usually it melts at 100 degrees centigrade and solidifies at 40 degrees centigrade. So most bacteria cannot hydrolyze agar. So these medias are formulated to contain the essential requirements for growth. For example, they have the source of energy, they have the source of nitrogen, they have the vitamins and also minerals that are required for the growth of microorganisms. Then we have some microbiological media. This microbiological media also contain other chemicals such as buffers, and these buffers are responsible for keeping the pH stable. These buffers keep the pH stable. Then they are also indicators or selective agents. These agents suppress the growth of unwanted microorganisms. You know, when you try to grow microorganisms in maybe a petri dish, there are some other microorganisms that can also come in and they take advantage to grow. But for you to suppress them, then it means that you will require certain chemicals that are known as buffers that keep the pH stable. So this will prevent the growth of unwanted microorganisms. Microbiological media, this mostly come in form of powder form, and it has to be reconstituted by adding the concentrates of water, after which the media is sterilized. If you look at that diagram, We have test tubes that have the broth culture. Then we have the petri dish, which has the agar. So on the topmost petri dish, they are growth colonies forming units. It means that bacteria has started to grow. Microbial cultures. Microbial cultures, growth of microorganisms and growth requirements. Microbiological growth media. These are found, for example, in the petri dish. The petri dish has a larger diameter. When you see the first one on the left hand side, it has a larger diameter and it has a shallow rim. The base is smaller and deeper. Okay, so if you want to use this petri dish, the base should be labeled with the details of the medium, the date, and the source of inoculation, or which microorganism have you put there. Then there are smaller spacers. These smaller spacers allow air to enter because we said that some of the microorganisms may need oxygen to grow. Then we have the normal use one, then we have the inverted petri dish. So inverting the plates during inoculation prevents condensation falling on the surface of the media, because condensation can result into some microorganisms spreading or streaming on the surface. Let's look at the liquid media or the broth. Let's look at the solid agar. We have also the liquid media, remember the solid agar. Then we have the defined and undefined media. So defined media has known quantities of all ingredients. For example, it has all the trace elements, the vitamins, the carbon source and the nitrogen source. And remember, we said that the major source of energy is from the carbon sources, which will give glucose or glycerol. Then the main nitrogen sources are ammonium salts, air, or nitrates. Then we have the undefined media, basal complex. This contains some complex ingredients, for example, yeast extract, casein, hydrolysate, blood. This consists of a mixture of various compounds in unknown proportions. Examples of undefined media, we have the nutrient media, we have the brain heart infusion agar, then we have blood agar. Nutrient media, undefined media. The nutrient media or undefined media, this contains a source of amino acids and nitrogen. The supply of this can be from the beef or yeast extract. Amino acid source contains a variety of compounds with the exact composition being unknown. So the media contains all the elements that most bacteria require for its growth. Okay? And it is non-selective. It is non-selective. And it's used for the cultivation, it's used for the general cultivation and maintenance of bacteria collections. Let's look at this slide, which has the types of microbial growth media. For example, on the right hand side, we have a column which has the component. The middle column has the grams per liter. Then the column on the right hand side has the role. For example, Lab Lemco powder, 1 gram per deciliter. What is its role? This Lemco powder is got from meat extracts, and it is a source of nitrogen, vitamins, and minerals for microorganisms to grow. Then we have another component, the yeast extract, which is also in grams per deciliter. This yeast extract is got from yeast, and it acts as a source of nitrogen, vitamins, and minerals for microorganisms. Then we have the peptone, which is in form of 5 grams per deciliter. Mostly its role is enzymatic digestion of meat, and is a source of nitrogen, vitamins, and minerals. Then we have the sodium chloride in form of 5 grams per deciliter. Sodium chloride will act in controlling the osmotic pressure. Then we have the agar, which is in form of 15 grams per liter. Agar, its work, it acts as a solidifying agent. Then we have the pH with the buffers 7.4 plus or minus 0.2. Its role is relatively neutral pH, and it is non-selective. Differential media stroke diagnostic media. Differential media stroke diagnostic media. These are used for differentiating between different bacteria or microorganisms growing in the same media by using an identification marker for a specific type of microorganism. You can be growing microorganisms in the laboratory, and if you want to identify the different bacteria or the different microorganisms that are growing on the same media at the same time, it means you are going to use an identification marker for a specific type of microorganism. So groups of microorganisms can be differentiated, or they can be separated by their ability to utilize the nutrients or by their form of growth. Examples of differential media, this include blood agar, mannitol salt, eosin methylene blue, mark conke, chocolate agar. Examples of blood agar contains mammalian blood, and this blood has been got from the sheep or the horse, and usually it is at 5 to 10%. Remember, mammalian blood, specifically the sheep or the horse, and not human being, not that. So these blood agars are used to isolate fastidious organisms and detect hemolytic activity. Okay, they can also be used to separate microorganisms on the basis of the ability to lyse red blood cells and also utilize hemoglobin. When we talk of ability to lyse red blood cells, it means the ability to break down the red blood cells. Thank you for listening and have a blessed day.